Hi, this is Mr. McLean, and in this video, I want to take a look at the concept of passive transport, and we'll look at some of the different ideas that are associated with that. So, one of the things to keep in mind with this chapter is that we're going to be looking at how things move across a selectively permeable membrane. So our plasma membrane is selectively permeable, also referred to as semi-permeable, meaning that some things are going to be able to pass through and other things are going to be restricted. And when we start taking a look at factors that influence whether or not something's permeable, a couple of the big things that would jump out at you is the nature of the molecule. So for example, things that are going to be hydrophobic, nonpolar, small molecules, things like carbon dioxide and oxygen gas, they're going to move across the membrane very easily. They're not going to have any issues, and you're going to be able to see a pretty significant rate of movement. If you look at some of the things that don't move across the membrane very easily, they deal with molecules that are polar or ions, in the sense of things that are charged. So when we move into looking at some of those structures or some of those molecules that are not going to easily move across, that's where we start to see what we refer to as facilitated diffusion. And the key thing to remember is facilitated diffusion is passive. Okay, facilitated diffusion is passive. So the same thing that's driving our normal simple diffusion or osmosis is a concentration gradient, and that's true with facilitated diffusion. So the big difference is whenever you have facilitated diffusion, you're talking about molecules that are either hydrophilic or they're so big that they're going to get resistance. Typically, they're things that are hydrophilic, polar, and they're going to struggle to get across. So a classic example of that is we talk extensively about water moving across the membrane, but water we know is very polar. So the aquaporons allow for sort of a safe passageway by creating this tunnel where the water molecules can move very aggressively across that. And when we look at those examples of things that are going to follow that type of movement, or environments where we have a lot of aquaporons, we're talking about things like our kidneys and being able to reabsorb massive amounts of water. So I'll talk more about that as we kind of move through here, but section 3 talks about what passive transport is. So it starts off with the simple diffusion concept, and that is molecules are constantly in motion and they want to move to an area where they can spread out to the available space. And if we have two environments that are separated, the environment eventually will get to a point where there's no net movement. And should that occur, we are then at what we refer to as a dynamic equilibrium. So if we look at these diagrams, let's start up here first with just the top half of this diagram. Initially, we have created an environment where we have a concentration gradient. All right, so please be clear that you understand what a concentration gradient is. My environment on this side appears to be 100% water and 0% solutes, whereas over here I have some concentration of solute. So we'll make it simple. We'll say this side has 90% water and 10% solute. So the concentration gradient exists because we have more solute on the left than we do on the right. And what we're going to notice is if this was not allowing the movement of the molecules of dye, then we would have a strictly only osmosis type of movement. But because there's permeability, so in other words, the way this is shown, you got spaces here where these molecules are going to be able to go across there. So we see net diffusion taking place, and then eventually we reach dynamic equilibrium. So when we look at dynamic equilibrium in this case, what we're noticing is the molecules have not stopped moving, it's just that there's no net movement. And that's the natural tendency. The other thing to keep in mind, when we look at the second scenario, I have two different types of molecules, and they are both able to move across the membrane because of the permeability, and they each are going to diffuse down their own concentration gradient. So it's really critical that you keep that in mind, that every substance has its own gradient. Everything will move until it reaches its own dynamic equilibrium. So think about the example we talked about in class, where you have the gas exchange between the oxygen and the carbon dioxide that's taking place in your lungs. So you're going to see a definite movement down the gradient. Now, when we move on to the concept of osmosis, that is selective, or that is the movement of water. That's what makes it different across a membrane that is permeable to only certain substances. So this is really critical. Water will move or diffuse across a membrane from areas of low solute concentration to areas of high solute concentration. So we go to the YouTube, and I can promise you there will be some questions on our test that refer to the YouTube configuration. So in this osmotic tube, we have an environment at time 0 that has more solute. So we have a very concentrated environment on the right side, and on the left side we have less solute. So we're going to assume that this membrane, in fact, it even tells us this, that the membrane is going to be permeable to water. So water's going across, but these sugar molecules are not going across. So it's going to be selectively permeable to the water molecules, not the sugar. So the only thing that we're able to see move across the membrane is water. And the water, if we go once again to make simple numbers, let's just make this real simple. Let's say this side has 95% water, 5% solute. And then this side over here was, let's say 90% water, 10% solute. So there's two ways you can look at it, and both of them are going to lead you to the same conclusion. If you look at it from the standpoint of where their solute's at, the solute is greater on the right side. And solute has a tendency to pull water towards it. The other way to look at it is if you look at it kind of from the standpoint of a ramp, and you got 95% here and you got 90% here in regards to water, it's going to go from 95 to 90. It's going to go down the ramp. So in this case here, our water moves towards this side, and because these molecules of sugar are not able to move across, we physically are going to observe this side rise up, this side sink down. The other thing to keep in mind, that these are open containers. So that tells me that there is no atmospheric interference, really. It's going to be a balance, and the pressure potential is not going to be a factor in here. So 4 words, or 3 words really, that you need to be familiar with in regards to the tonicity. So tonicity is really talking about the solution itself, if it's going to gain or lose. So we define our solutions as either being isotonic. So isotonic is equal, okay? You have the same on both sides of that membrane, and as a result, you're going to have no net water movement. All right, so whenever something reaches kind of dynamic equilibrium, or if it's just set up that way, we have isotonic. If we have a scenario where something is hypertonic, what we're going to find is you're going to have solute concentration is greater than inside the cell. So if I were to set up my beaker with my cell, and let's say that this cell was 80% water, and my environment out here was 75% water, and the remaining portion of it was 25% solute, then in that case right there, I would have a hypertonic environment on the outside. So if I have more solute, it's hyper, and therefore the water is going to want to go out, and it's going to want to go into that beaker. If it's hypo, it's going to be exactly the opposite. So if I were to take something and let's just make it here, I'll put myself a blank slide in here. If I take and I have a similar situation, and in this one I have my cell. Let's make my cell 80% water, 20% solute. And I put it into an environment that is 90% water, 10% total solute. So in this case right here, I have 90 being greater than 80. So just like your ramp, 90 will flow to 80. So water is going to go in. That would be hypotonic. Okay, so hypotonic creates that kind of situation. So if we do a quick comparison between plant and animal cells, we notice that animal cells respond differently to these three scenarios. For an animal cell, we want this right here. That's a red blood cell that is under isotonic conditions, and you're going to notice that it maintains normal shape and normal functionality. If we put that animal cell in a hypotonic environment, the water is going to rush inside of it, and it's going to have lysis. So we call that cytolysis. Cytolysis. And then over here, the opposite happens. If we put in a hypertonic, so if you were ever lost at sea and you were to consume salt water, that would cause further removal of fluid, and that would be a big problem this way. So you'd have the shriveling. For plants under isotonic conditions become flaccid. Okay, so that's not the ideal. That would be kind of the plant is somewhat limp, and the reason is there's not enough water filling up that central vacuole. So the pressure that would normally push out against this plasma membrane and then eventually against the cell wall is not significant enough. Plants want to be in this condition right here. They want turgor pressure. So when you think about it, you go into a grocery store and they have vegetables that are being sort of misted with fresh water, or if you take a rose and you put it into a little vase with fresh water in it, you're keeping the turgor pressure up by creating a here it is right there, a hypotonic environment. Worst case scenario for a plant is this last one where you get plasmolysis, and that's in hypertonic, and basically so much of the water is leaving that you have now compressed that cell because that vacuole has kind of left all its water out, and there's a possibility that you're going to tear that membrane. So that is plasmolysis. Other organisms, such as the Paramecium, they use different methods of osmoregulation. Because they live in a freshwater environment, this is a contractile vacuole. It's filling up with water, and then as it fills up, it expels its water to try to maintain osmotic balance. Okay? So maintaining osmotic balance is definitely one of the most critical things that we can look at. All right, and then the last thing that I have in this section is talking about the idea of facilitated diffusion, which I've already looked at. And we have two general methods of facilitated. The top one is showing us a channel protein, and a channel protein creates exactly that. It's a very general channel. These are typically going to have a gate on them, and they're going to be ion channels in many cases, and we'll see more of that in an upcoming section. The other picture that we have right here is a carrier protein. Now, both of these images are showing us going from high to low. So anytime you go from high concentration to low concentration, you're dealing with passive transport. And the big difference between these two is that this one involves a conformational shape change that expels the substance onto the other side of the membrane, and neither of these things are going to use energy. So these are both examples of passive transport. Okay, so I'll introduce another video where we take a look at some of the methods of active transport. Hopefully this helps clear some things up. Thank you.